Hi everyone. Today we're going to learn about BDNA. So, uh, in this particular diagram, you must be able to see that there are two polynucleotides, wrapped around one another and in a very helical manner. So, this is actually the BDNA, and there are many types of other DNA. For example, there is aDNA, and then there is zDNA as well. But then the most commonly found DNA in the human genome is bDNA. So we are currently studying about it. So in this particular video, I'm going to talk about the diameter, how many base pairs are going to be in one turn, what will be the rotation of the base pair. So all these things we're going to discuss in this particular video. So let me first try to tell you what exactly is this figure trying to tell you. So as you can see that the DNA is present in a right-handed manner, right? So it is present in a right-handed manner, and it is going in the direction of an anti-clockwise direction. So whenever we talk about clockwise direction, we know that this is the clockwise direction, and when we talk about anti-clockwise, we go this way, right? So as you can see that the DNA is going this anti-clockwise direction. Now this anti-clockwise direction. So now when we talk about DNA, this is actually the polynucleotide. This is the sugar and the phosphate backbone, and then there are bases which are present out here. So these are the bases which are present out here, and then the bases, the nitrogenous bases, form hydrogen bonds between them. So this diagram is showing the bases and the hydrogen bond, and this particular red structure is actually the backbone, that is the sugar and the phosphate, right? And the sugar which is present in this particular DNA is deoxyribose sugar, which does not have an oxygen in the second prime carbon, right? Now let me just talk about the structure of DNA. So when we see this part, this part, this is actually the diameter of the DNA. Right? This is the diameter of the DNA. Now let us go into other details. When we see these things, there is like a dent which is present. There is a larger dent, a very wide dent, and then there is a narrow dent, right? So these dents are actually known as grooves. But then when we talk about their names in terms of their wideness, we term this groove, this particular groove, which is like gone inside and it's very wide, we're going to call it as we're going to call it as major groove. And then as you can see, there is also a smaller groove present over here. We're going to call this as minor groove. Okay. Now if I want to see the turn that has been made by this bDNA, then I'm going to take consideration of this particular major groove to this particular major groove, because this is also a major groove, right? This is a minor groove, this is minor groove, minor groove, minor groove, minor groove. These all are major groove, major, 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 and then major, right? So if I have to if I have to say that this DNA is turning, like it has completed one turn, then I'm going to take this major groove and then this major groove, and I'm going to say that this is the complete turn of that particular DNA. Or I can also take this minor groove, and then this minor groove. This will also be considered as a turn, you know. So this to this will be turned as pitch, or you can say turn. Or we could also say in terms of this to this could also be termed as pitch or turn, right? Now I'm going to remove this because I have to write down certain details. So now when I talk about the helix of this particular bDNA, I'm going to say that this is an anti-clockwise. I'm sorry, right-handed. First let me write down right-handed. This is actually a right-handed helix which is anti-clockwise in direction, right? When I explained you what exactly the diameter of the bDNA is. When I talk about the calculation or the measurement, it's going to be 20 angstrom. Or you can also write 2 nanometer. So nanometer is like a smaller unit as compared to the angstrom, right? Not a smaller unit, but we write it like angstrom is a smaller unit and nanometer is like the larger unit. Okay, so now when we talk about the major groove, as you could see, the major groove was quite wide, right? It was wide, and then minor groove was narrow. Now why am I saying this is narrow and wide? Because this is definitely very wide, this is definitely very wide, right? And this is very narrow, right? Okay, so now when I talk about the pitch, you know, the pitch is around 34 angstrom. So angstrom, okay. So the pitch which I discussed, this, the measurement of this is 34 angstrom. Now when I talk about base pair per turn, it's going to be 10.4 in number, base pair per turn. Now what do you mean by like base pair per turn? That means this is particularly one turn, right? This is one turn. Now how many bases are present in one turn? That means I'm going to count all the bases which are present from, let me take another pencil, this one. I'm going to take this 2, 3, no, this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then 0.4, right? So I'm going to take all these bases which are present in particularly one turn. So that will be my base pair per turn. Okay. So that is going to be approximately 10.4. So in one turn there are almost 10.4 base pairs present. Now let us see another one, rise per base pair. Now rise per base pair means like if there are two base pair present, right? If, for example, if there are two base pairs present, then what exactly is the height of this what is exactly the height between two base pairs? That means one base pair is present out here and another base pair is present out here. So what exactly the height? So can I say that I can divide this pitch and then 10.4? Let me just divide and then I'll come back. Okay, so 34 divided by 10.4, that will be approximately 3.2 angstrom. So we can say that 3.2 angstrom, right? If I talk about in nanometer, that will be 0.32 nanometer, right? Okay, so that is the rise per base pair. That means the distance between two base pair, two consecutive base pairs, right? So if I just have to show you out here, you know, these two are base pairs, right? I'm just gonna take another color. This is like this one. Okay, so these two are base pairs. So what exactly is the distance between these two base pairs is known as rise per base pair, right? Okay, now let us go to the last one, that is rotation per base pair. Now imagine, if you are like, if you are just walking in a spiral staircase, and then you are moving each stair, and imagine that particular stair to be like the base pair of the DNA, because as you can see out here, this looks like a stair, right? This looks like a stair. And imagine like you are just stepping on each stair, and you are just moving towards the spiral, towards the upward direction of the spiral staircase. So when you're moving upward, you obviously step on another staircase, and then you also turn your body. When you're about to step on the another staircase, you also turn your body to one particular angle, right? So you are rotating to some point. So that is exactly the rotation per base pair. So the rotation per base pair would be approximately 33 degree to 34 degree. That means each time you are stepping on a new base pair, your body is rotating a little bit, tilting a little bit, and then that is exactly the degree of the rotation. So I hope you are able to understand this particular thing. So this is the bDNA. This is commonly found in the human genome, and the diameter is 20 Armstrong. There is major groove, minor groove. The major groove is wider, minor groove is narrower. The pitch is that one turn, and then in one pitch there are almost 10.4 base pairs present. And then there is also the distance, or yes, the distance between two consecutive base pairs, that is called the rise per base pair. And then also there is presence of rotation every time we move towards the next staircase, like the base pairs. Like if we move towards the base pairs, then we have a specific rotation, that is rotation per base pair. So I hope you're able to understand this video, and if you did so, please like, share and subscribe, and thank you so much.